So there's a new vaccine that is generating a lot of interest. Let's get into the science of what this is and go over the study that was just released today in this video. What is up? Welcome back to the channel. I'm Zach with Dr. Eyeball MD. If you are not already subscribed to the channel, go ahead and do so down below. And if you do find any value from this video, go ahead and like the video. What we're looking at in this video is a paper that was just published today, July 14th, in the New England Journal of Medicine that goes over the phase 1 clinical trial of a new vaccine that has been developed by a company called Moderna that is showing some promising results. So this phase 1 clinical trial was funded by the National Institute of Allergy and Infectious Disease, as well as Kaiser in Seattle. And this vaccine was co-developed with researchers at Moderna in Massachusetts. The vaccine was developed in record time, taking only 66 days for trials to start after the genome of the virus was published on January 10th, 2020. So what is this vaccine? It is called mRNA-1273. That is the name of the vaccine. So what is mRNA? It's kind of the intermediary step that gets you from DNA in your genes to the end product, which is proteins. The mRNA kind of is in the middle, and it takes the message from the genes and helps translate them into proteins. It's what's read from the DNA so that you can make proteins. This mRNA vaccine codes for a spike protein on the virus that helps the virus enter into the cells. So essentially what is happening is when the vaccine gets into cells, that messenger RNA is translated into that spike protein, which serves as the antigen, and that antigen stimulates an immune response so that the body creates antibodies. That way, when you get infected with the real virus, if you were to get infected with the real virus, you would have those antibodies that would be ready to attack. So it's essentially just a way to kind of input a genetic signal into the host cell, get the host to produce antigen, and then the body can elicit an immune response to that antigen. That's kind of the idea behind how this vaccine is working. Let's talk about the trial itself. So the vaccine itself is kind of a combination of this mRNA and a lipid nanoparticle. So they're kind of fused together. So in this study, because it's a phase 1 clinical trial, the researchers were evaluating the safety and the immunogenicity of this vaccine. There are different phases of clinical trials, phase 1, phase 2, and phase 3. And this is a phase 1 trial, and the primary goal of phase 1 trials is to evaluate the safety of the drug in question and also the dosage. If the drug passes through phase 1, it can move into phase 2 clinical trials. Typically, a phase 1 clinical trial will have somewhere between 20 and 100 participants. In this study, they had 45 participants. These were healthy adults between the ages of 18 and 55 years old, and they received the vaccine in the shoulder as an injection, and then 4 weeks later, a second vaccine. They tested 3 different dosages, 25, 100, and 250 microgram dosages. They reported their findings all the way through day 57 of the trial, and then they are continuing to gather research and plan to continue gathering data on these patients up to a year out from the second vaccine. Because one of the things we're looking at in a phase 1 clinical trial is side effects, they had the participants record any side effects that they might have a week after the vaccine was given. Side effects primarily consisted of fatigue, chills, headache, myalgias, and fever, in addition to pain at the injection site. At day 15 after the administration of the first dose of the vaccine, all the participants in the study had seroconverted, meaning they were all producing antibodies to the glycoprotein, the antigen that is essentially the vaccine that is stimulating antibody production, meaning all the participants were now making antibodies, which was the goal of the vaccine. There was a dose-dependent response, meaning the higher concentration vaccines produced a more robust antibody response. The antibody response was even greater than the control group, and the control group in this study was 41 serum samples of convalescent serum, which is basically just serum that is from a patient that has been exposed to the virus already. So they already have antibodies to the virus. So those were the control group, the serum samples that already have the antibody because they've been exposed to the virus. And this vaccine was producing an antibody response that was even greater than those convalescent serum control samples. The way that they tested to see if the immune response that was being generated by this vaccine was an adequate response was by testing the serum from these patients where they had generated these antibodies to the vaccine. They tested against, one, they tested against pseudovirions, and two, they tested against wild-type virus to see if it could suppress the wild-type virus. In testing the pseudovirus, or a virus-like particle, after the administration of the first vaccine, less than half of the participants were able to neutralize the pseudovirus. But after the second vaccine, after the second dose at that 4-week period, all of the participants were able to neutralize that pseudovirus, or pseudovirion. The other thing they looked at was to see if the serum from these patients treated with the vaccine could neutralize wild-type virus, at least 80% of it. And by day 43 after the administration of the first vaccine, all the patients were able to neutralize at least 80% or greater of the wild-type virus. Now, because this vaccine is being tested on healthy patients that are not actually infected with the virus, so these were two kind of functional biomarkers to see if this vaccine was generating an antibody response that was good enough to be able to kill the virus if it were to be encountered in a real-world situation in a person. Because obviously they weren't infecting these participants with the virus. They were just seeing if they could generate an antibody response, and then taking that serum and seeing if that serum could then have a response at neutralizing the virus. In short, what this means is that this vaccine was inducing a good antibody response that was able to neutralize virus, which is ultimately the goal. It's pretty amazing what these researchers were able to accomplish within just a couple months, something that would normally take years to accomplish in terms of drug testing and phase 1 clinical trials. They accomplished it very rapidly, and part of that is because this company, Moderna, already had experience with mRNA-based vaccines, and so they already had a platform where they could essentially tailor it to the specific virus. This is just a preliminary report that was published in the New England Journal of Medicine. It kind of takes us out to day 57 of this clinical trial, but they plan to continue following these patients for the next year or so and continue taking serum samples to basically determine if this immunity is lasting, because some prior evidence on the initial SARS virus and the MERS virus showed that the immunity may not be long-lasting, especially in milder cases. So they're going to follow up for a long period of time to see if this immunity is lasting and to what extent. Phase 2 clinical trials are already moving forward, looking at the kind of medium concentration dose and the lower concentration, the 50 and the 100 concentrations. There are already phase 2 clinical trials underway with about 600 participants, and then phase 3 clinical trials are expected to start in the summer of this year with enrollment of probably greater than about 30,000 patients in that phase 3 clinical trial. So while it may seem like it's taking a long time to develop this vaccine development is something that normally takes years and years, so it is actually being super fast-tracked. And while we would like it to be done right away and even quicker, it is important to go through these steps to make sure these things work, to evaluate the safety profile of these vaccines, and make sure that the dosages that are being used are not toxic and that are safe. So it's important to follow those steps, but fortunately the researchers are definitely fast-tracking this and getting it done at record pace. Okay, so to summarize and kind of what to take away from this clinical trial, because it is spurring a lot of excitement, not just for the vaccine itself, but also it's spurring a lot of economic excitement for this company, Moderna, that is producing this vaccine. The basic takeaway here is that they have developed an mRNA-based vaccine that enters into the cell, helps your own cell produce the antigen that allows a good antibody response to be generated. And from this phase 1 clinical trial, it is looking like it's a pretty robust response, maybe even more robust than the antibody response from the convalescent serum samples, meaning maybe even more of a robust antibody response than the antibody response that you get from actual wild type infection of the virus. So we will see how this pans out in the phase 2 and phase 3 clinical trials, so be on the lookout for those. I will link to the New England Journal of Medicine paper in the description of this video if you want to go read it for yourself. Look at some of the tables and stuff that they have included. It's pretty good. And anyways, I hope that was helpful. I hope this kind of clarified what this new vaccine is hoping to accomplish and why there's a little bit of excitement around it right now, just because it is generating a good antibody response and could be a potential way to prevent people from getting this in the future. So we will see. Anyways, if you guys liked the video, leave a like down below. Subscribe to my channel. I'm Zach with Dr. Eyeball MD. I will see you guys in the next video.